Using a grasshopper as our model, let's briefly explain the organization of the insect central nervous system. The insect central nervous system consists of a brain that is connected by a pair of nerves that pass around the esophagus and connect to a nerve cord on the ventral side of the insect body. The ventral nerve cord generally consists of a ganglion in each segment with the ganglia interconnected by paired nerves. Ganglia are thickened regions of the nervous system that contain the cell bodies of the neurons. Nerves are bundles of axons from many individual neurons. Here is a hair mechanoreceptor on the leg of the grasshopper. Mechanoreceptors detect pressure stimuli that cause the hair to bend. This fade-in shows the external hairs attached to a sensory neuron that extends its axon through a lateral nerve and into a segmental ganglion. The hair receptor monitors the intensity of the pressure stimulus based on the amount of bending of the hair, which pulls on the sensory dendrite attached to the base of the hair. A weak stimulus causes a small depolarization of the dendrite and a low frequency of the action potentials in the axon of the sensory neuron. A stronger stimulus causes a greater depolarization of the dendrite and a higher frequency of nerve impulses in the axon. Lastly, a strong stimulus causes an extreme bending of the hair and a strong depolarization of the dendrite, resulting in a high frequency of nerve impulses in the axon. The action potentials in the axon of the sensory neuron are transmitted to a secondary ganglionic neuron in the ganglion of the ventral nerve cord. Here is a cutaway view of the hair receptor, its sensory neuron, and the path of the sensory axon from the leg through a lateral nerve into a ganglion of the ventral nerve cord. The next scene will demonstrate how the synapse converts the action potential frequency from the sensory neuron into information within the ganglion of the ventral nerve cord.